Thank you very much. And we'll now have the final presentation in this panel by Dr. Britt.
Okay, thank you for allowing me to give this presentation. Again, I echo Mike's comments. I'm sorry I tried to make it, but the airlines were not cooperative. Okay. I presume we're connected.
Yes. Yes, go ahead, Bill.
I'm sorry. Yes, and so I'm going to speak today about congenital viral infections and try to draw some parallels between rubella and CMV. Really not much discussion about Zika because little is known, if any is known. Next slide, please. Congenital viral infections are relatively common, and they're defined as a transplacental acquisition of a virus in contrast to perinatal infections, which you see on the right-hand side, which includes some of the hepatitis viruses and as well as some of the herpes viruses. Congenital infections are often associated with central nervous system disease in contrast to perinatal infections with the exception of herpes simplex virus. So these are rates of congenital infections in Brazil, drawn from studies that have been published and studies that we've carried out in Brazil. And as you can see, cytomegalovirus is relatively common, and that rate is somewhere between 6 and 10 per thousand live births, which in Brazil calculates to about 20 to 30,000 babies a year are born with this congenital virus infection. Next slide, please. So with the frequency of these congenital infections and today's technologies, why don't we know more about the mechanisms of transmission and disease with these agents? Well, primarily because we're really dealing with three systems simultaneously when we try to study congenital viral infections. We're dealing with the pregnant woman, the placenta, and the fetus. Each is a complex biologic system. Each is developmentally regulated during pregnancy, and thus it's difficult to study one system in isolation from the other three. And I think this has led to a lot of the difficulties in really understanding the pathogenesis of many congenital viral infections. Next slide, please. However, there have been some systems that are a little bit easier to understand. One was rubella. And rubella virus resulting congenital rubella syndrome is shown here. And what we learned early on was adaptive immunity could block viremia, block placental infection, and block fetal infection, as shown in the third line. Thus, achieving adaptive immunity would actually prevent this syndrome. And this was, I think, reflected early on in the UK when they studied the incidence of rubella and congenital rubella. And although this is not a well-done slide, you can see that the red bars indicate the disease activity of rubella. And when the disease activity of rubella fell, the incidence of congenital rubella increased because of the increase in number of young women that were susceptible to rubella infection. More recently, studies in Asia have looked at the rates of congenital rubella syndrome versus a seroprevalence in the population. As you can see, when the seroprevalence reaches levels of about 80 or 85%, there's no congenital rubella syndrome, effectively eliminating this as a congenital infection. Indeed, the 85% seropositivity level was a vaccine uptake target used in the early deployment of rubella vaccines. Next slide, please. In contrast, cytomegalovirus has a much more complex epidemiology. Maternal immunity is clearly important because we know that if we In women who have a depressed immune system, such as women with uncontrolled HIV infections, the incidence of congenital infection in their offspring is much higher. However, we don't know how adaptive immunity works. There have been reports of adaptive immunity interfering with viremia and with placental infection and with fetal infection. However, none of these have been shown to completely block the transmission of this virus to the fetus. In addition, adaptive immunity in CMV shows somewhat of a paradox. As the rates of seroimmunity to this virus, that is, as the number of women with exposure to this virus increases, we see increased rates of congenital CMV infection. And this is in contrast to rubella, which is shown in the inset on the lower right-hand corner. So the more women in the population that are immune to CMV, the more likely it is to see congenital viral infections. And again, this is reflected in that we see a much higher yield of congenital infections from populations with non-primary, that is, women with immunity, and than those who acquire CMV as a primary infection during pregnancy, about a 3 to 4-fold increase. Next slide, please. And why is this? Well, without going through this details on this slide, which are difficult to read, I can see, we do see superinfection. That is, women with established immunity to CMV can be reinfected with new strains of CMV during pregnancy. And this was first detected using an assay to define serotypes of CMV. More recently, when next-generation sequencing has been done on CMV isolates, we see that CMV actually circulates as a quasi-species. Such as that are complex mixtures of genetically diverse viruses in most individuals, therefore providing at least a reservoir of viruses that could reinfect these women. As you can see in the bottom panel, this diversity approaches that of dengue virus. Next slide, please. So the next step of infection is to cross the placenta, and as Mike alluded to, this is the source, this is the site where the virus enters the fetal circulation. And this is a very complex organ, as most in the audience realize, and it has a very unique structure in primates and some rodents, in that the villus structure sits in a pool of maternal blood, as illustrated in the light green circle. This then enables any infectious agent that could be present in maternal blood easy access to the fetal circulation, as this in some periods of gestation only are represented by a single cell layer thickness. Next slide, please. And this is, again, illustrated by the placental development that could facilitate virus transmission. Placental blood flow is incredible. By the mid to second trimester, 30% of the maternal blood flow passes through the placenta, therefore providing a real ample opportunity for any blood-borne pathogen to enter this space. As I mentioned, during the mid to second trimester, the covering of the villus thins, such that the only thing that separates maternal blood from the fetal endothelium is the base of the membrane of the syncytia trophoblast. So, again, an easy access to the fetal circulation. In addition, there are a variety of cells in the villus stroma: endothelial cells from the fetus, stromal fibroblasts, cytotrophoblasts, and macrophages that are in the stroma that could provide permissive cells for virus replication and amplification for the fetus. Next slide, please. In addition, we shouldn't forget that the major function of the placenta is to provide nutrients and oxygen to the developing fetus. And viruses that infect the placenta can certainly interrupt this function. In the top panel, it's shown that CMV can infect trophoblast progenitor cells of the placenta, limiting their self-renewal capacity, therefore limiting the number of functional units available in that particular placenta. In addition, other investigators have shown that CMV can infect the placenta and result in a thrombotic vasculopathy, again, which decreases the function of these functional units of the placenta. In both cases, these interrupt nutrient and oxygen supply to the fetus and can result to damage in the central nervous system, damage in other organ systems that probably contribute to some of the syndromes we see with congenital viral infections. Next slide, please. So what are the diseases once the infection occurs in the fetus that we see? And I'll focus only on the central nervous system because there are clearly other organ systems involved. Most of these are self-limited and rarely have long-term consequences, with the possible exception of the cardiac defects and rubella. So the clinical findings we see are microcephaly, neurologic deficits such as seizures, deficits in perception such as sight and hearing, and this is very important. For instance, congenital cytomegalovirus probably accounts for about 25% of all the hearing loss in children in the United States. There are neurodevelopmental delays, laboratory imaging findings, structural malformations, and this is basically developmental stage specific of the brain when the insult occurred, calcification, vascular damage, loss of neuronal mass, and evidence of CNS inflammation. But as you can see, these are all relatively nonspecific responses to injury to the brain, and again, dependent on the developmental stage of the brain. Next slide, please. And this is illustrated. This is a photograph of provided to me by an investigator in Brazil showing an infant with microcephaly that was associated with a maternal Zika infection. The CT scan on the right shows the calcifications in the brain, as you can see by the white spots along the ventricles, the large ventricles indicating loss of the cortex in this part of the brain. Next slide, please. This is a child with congenital CMV. This child also has microcephaly and some of the same stigmata that the patient in Brazil had. And the right is a CT scan from another child with congenital CMV, and you can see the calcifications, the large ventricles, and the loss of the cortex. So again, these are very nonspecific findings and provide us little insight into the mechanisms of disease caused by these specific agents. Next slide, please. So what are the direct postulated mechanisms of disease, direct virus-mediated damage to the developing CNS? There could be loss of neural progenitor cells. Clearly, these viruses have been shown to infect neural progenitor cells and cause damage or loss. Importantly, they could infect the supporting vasculature in the CNS, and as a result of any loss of vasculature, certainly you would lose the parenchyma of the CNS. They could result in altered cellular positioning of cells, altered connectivity within the CNS, increasing apoptosis if these connections are not made, and virus-induced alterations in neurotransmitter response, again leading to neural dysfunction. However, all of these may be operating simultaneously, and they provide often a very variable phenotype, even under controlled experimental conditions. In the bottom panel, these are brains taken from rhesus macaque fetuses that were inoculated transplacentally in the second trimester. They were not inoculated in the brain, they were inoculated in the peritoneum. The virus spread to the brain and caused these brain defects. You can see the brain from the control animal on the left with the normal structure of the cerebral cortex. The animal on the far right, number 1, you can see has lissencephaly with loss of all structure of the cortex. A coronal section of that brain shows the ventriculomegaly and the loss of the cortex. In the middle there, one of the fetus inoculated under the same condition shows another pattern of pathology in the sense of loss of certain areas of the brain, focal infections, and focal loss of the cortex. So again, the pathology is probably dependent on the magnitude of each of these possible mechanisms of disease. Next slide, please. And finally, as Mike alluded to, the possibility of host inflammation as a cause of some of the disease we see in infants with congenital CMV shouldn't be overlooked. And this is data that we've generated using a newborn mouse model of congenital CMV infection, in which we use murine CMV to infect newborn animals peripherally, not intracranially. And we know that about 8 days after infection, we see marked changes in their central nervous and their brain development. And on the right, you can see the cerebellum from one of these infected animals. It's much smaller than the controls. The foliation, that is the complexity of the cerebellum is much reduced in this animal. This is not associated with widespread virus infection in the cerebellum, but with widespread inflammation and induction of a variety of inflammatory genes. Below in the panel on the left, you can see what happens when we treat these animals with steroids, and that's shown on the right side of the panel. Those are virus-infected animals. The white histogram shows a vehicle-treated animals versus those treated with steroids. And as you can see, we can normalize the cerebellar area and structure by just treatment with steroids. And on the right-hand side, you can see that some of the genes in the cerebellums that are developmentally regulated are also normalized by treatment with these steroids, and those are shown in the shaded bars on the right sides of each one of the columns. So host inflammation probably plays a significant role in the damage we see in congenital infections. So last slide, please. Last slide. So the resource priorities are really, and I think they echo what's been noted in the earlier talks in this session and the earlier talks this morning. We really need to generate comprehensive and definitive natural history studies of congenital and perinatal Zika virus infections. These will likely require new diagnostics, prospective studies, newborn screening, and long-term follow-up. And I think this is very important because we really don't know the spectrum of disease with this virus. We see the cases of microcephaly, but there could be subclinical infections that end up contributing to long-term morbidity in these patients, be it loss of hearing, for instance, or impairment in vision. So these need to be well done and very comprehensive studies. Secondly, these studies will help us inform the development of informative animal models that recapitulate really key aspects of the human infection and allow us to design strategies and interventions to prevent these infections. Thank you.
Thank you.